Let's turn to another strategy, if I may say so. My main concern is to understand how the brain is developing. I've been working on it for 40 years, made some progress, and you'll see this is related to autism, and sometimes we may run into good surprises. The substantial problem, we touched upon that earlier, to develop a brain, you need a genetic program, but that's not enough. If I wanted to build this building, I would pick an architect, and then we would meet to talk about the works every week. Otherwise, the building wouldn't be straight. The question is, who organizes the works meeting? In biology, it's called a feedback. You have to feedback to check that the genetic program is underway as it should. Well, and the activity of the brain during development, that's the feedback. We start generating electrical activity very early on in life. This electrical activity in children is drastically different from what it is in adults. An immature brain is not a small adult brain. It abides by its own laws and rules and proteins and genes that do things different from what they do in adults. So I want to understand how activity is established during the development and how it's modified when you have a brain insult or any other disease. Simple example. Do you know when we open up our eyes during development? At the end of the first quarter of in utero gestation. You might say, well, we can't see anything at that stage, but if you record the retina at that age, the retina will generate an activity which is incompatible with eyesight. If I had that type of eye acuity, I wouldn't see anyone. It's too slow. But if you block that activity, the visual cortex will not develop correctly. So it's an activity that's pointless in the sensory fashion, but without it, the brain will not develop correctly. Did you get the nuance? So it is necessary, but it doesn't produce eyesight. You cannot see at that stage, but it's useful. Second example, ladies, if you've been pregnant, you know that a fetus, a baby, moves a lot in the womb. We've recorded these movements in premature babies and in rats and mice. If I do that, if you record my hand and motor cortex, it's the motor cortex that says you move your hand now, but not in them. If you record the cortex in premature babies and movements, the movements most often have a peripheral origin, meaning that these moves are generated by the spinal cord, the bone marrow, and they help to do all the wiring in the brain. It's the works feedback. You perceive the periphery wires up what should be wired up in the brain, and that's key to understand the rest. Now, on the basis of these two examples, we know that the periphery helps me to wire up my brain, and the genetic program is key, but it's not sufficient because a genetic program doesn't allow you to check that what was programmed was indeed achieved correctly. Now, the theory we developed with my colleague Spencer is called the checkpoint. The idea is that at each stage in the development, there is a control at the level of the cell to check that everything went all right. It could be shown under this form. It's not A that is true. It's not gene A that does function A, gene B doing function B. Yes, of course they do that, but that's not how it works. Gene A carries out a function A, electrical activity, which in turn will promote, facilitate, or hamper, block the expression of gene B. That changes the whole perspective, and it can be shown under that form. I don't have time to go deeper into that, but I'm currently about to prove a fundamental argument that I call neuroarchaeology. It's only normal I was born in Egypt. The idea is simple. If what I told you is right, what happens if a neuron doesn't do what the genetic program scheduled it to do? I'm currently proving that a neuron that should migrate from A to B and which does not migrate for any reason, Thomas' favorite genes, pesticides, stress, whatever the cause. But if it doesn't do it, the cell electrically will remain immature and it will not go from A to B. We are starting to collect evidence, and it's important because one day we'll be able to treat these patients with drugs that block the immature proteins, but not affecting adult cells, adult proteins. And that will be a revolution in neurobiology. So if there is a conflict between the program and its realization, the conflict will be expressed through delayed in the developmental sequence. And this has implications on the topics of concern to us. If the insult starts very early on, you'll have a whole cascade of events depending on the primary insult, and that will be manifested through networks generating apparent activities very early on in utero. Well, I can prove that with a genetic transfection type of approaches, but now let me shift to another topic based on a discovery we did with my friend Eric Lemonnier. It's the following. You know, it's a developmental disorder. You know that some genes play a role, the environment as well, but the GABA receptors, what's the GABA? Let's start with that. The GABA is the main mediator for brain inhibition. It's a chemical product. When released, it acts on a receptor. In the middle, you have a canal, which is patent to chlorine ions. Chlorine has a negative electrical load. What happens in adults, you have lots of chlorine outside, very little chlorine inside. And when you have chlorine, it inhibits because it's got a negative load. The potential is more negative. You have less discharge. Some 30 years ago, I discovered that in the immature brain, this is not true. You have lots of chlorine inside in a quiescent state, meaning that the chlorine will be released, will go out, and instead of inhibiting, it will excite. In a nutshell, that's the idea. The implications are huge. If you take Valium because you have anxiety, what Valium or pentobarbital or phenobarbital do is to increase the efficacy of GABAergic currents. So if you're in that situation, he leaves the microphone. We cannot hear. Okay, on the right, less anxiety, on the left, more anxiety. We lose the speaker, says the interpreter. No microphone. A pregnant woman taking Valium, the effect of Valium on the mother will not be the same as it is on the baby. Then we discovered that in brain pathologies, epilepsy, trauma, lesion of the spinal cord, what have you, something is such that chlorine accumulates inside again. So neurons, after being insulted, are young neurons. So chlorine initially is you have high level of chlorines in a young subject, and if you damage it, it goes back up. It's a major phenomenon in epilepsy. The kids, when you treat them with phenobarbital, it aggravates the seizure instead of blocking it. Now, the starting point here is relatively simple. Valium, as we said, may have paradoxical effects in autistic children. You understand, because you listen to what I said. If Valium has a paradoxical effect, it might mean, it's not an evidence, but it might mean that the chlorine level is high. If the chlorine level is high, well, here I must make a parenthesis. Chlorine is not only anions. It's crucial for several reasons. It regulates water. Chlorine regulates osmotic pressure. It's crucial. And regarding evolution, that's why chlorine was maintained at a high level since the beginning of evolution in seawater and so on. So chlorine is key in a number of functions. GABA receptors are the only mediator in which the polarity can change according to the activity. No other one does that. Why? Because if you excite the GABA receptor, you allocate the systems that control how what it does, and you'll get the answer. Now, I'm going to give you an example which is not relating to autism, but it's quite fascinating, and it's what convinced me that I was right in my assumptions. We measure in animals the intracellular chlorine levels. It's very difficult to achieve. I'll skip on the details. We're practically the only ones using that technique. So we measure intracellular chlorine in the cortex of mice. In utero, you have a very high chlorine level, and gradually it will go down. We expected that result. What we didn't expect, however, is that on the day of delivery, of birth, you have a drastic drop in intracellular chlorine level, so high that the chief editor of Science told me it's impossible thermodynamically. You cannot have such little chlorine in a cell. It's unheard of, incredible. I'm not Galileo, but I answered what Galileo pretty much answered. You know, the facts are here, so it's the theory that's wrong. So on the day of delivery, of birth, you have a drastic, dramatic drop in the chlorine levels. So what, you might say? And here, it's a bit like what Angela was saying. At the time of birth, labor, oxytocin plays a key role, and if you block oxytocin, you block that drop in chlorine. In a nutshell, oxytocin acts by reducing intracellular chlorine. That's how it acts. The pathway is not yet totally clear. I'll come back to that, but obviously chlorine plays a key role in what oxytocin does. And this is not a theory, because one thing that happens during birth that may happen when you have complications is a case of anoxia, a lack of oxygen in the brain. Which is a cause for the cause of many negative consequences. If you block oxytocin and you measure the resistance to anoxia, if you block oxytocin, the brain is much more sensitive to anoxia. Oxytocin, in other words, plays a neuroprotective role. It's a molecule that activates the uterus, triggers labor, and protects the neurons, the brain cells of the baby, from early anoxia. And that's not all. A paper was published in Sweden. They measured, I don't know how, the level of pain in babies right after birth. It's in Sweden. I'm sure they didn't torture them. They took some blood samples and measured the facial mimics, crying, reaction, and so on. Well, listen, babies born with a C-section are more pain sensitive than babies born normally. It's quite impressive. I'm not in favor of scheduled cesarean section, but anyway, a difference between women scheduled for C-section and the others, the difference is oxytocin. We tested that in rats, and we've proved in a binding way that in rats, the pain pathway, well, oxytocin acts by decreasing chlorine levels, and it has an anti-pain action, and this action is imitated by the diuretics. Chlorine is regulated by many things, dozens of systems. Here I kept it simple, no need to. You have the KCC1 carrier bringing chlorine in, and KCC2 that takes the chlorine out. This one, if you have a problem of diuresis or high blood pressure or high liquid level or edema, you will be given a diuretic. It will take the water out. And how does the diuretic act? It will block NKCC1, so chlorine will not go in. Chlorine and it will take the water and be released. This other gene exporting chlorine is very fragile. In epileptic tissue, it disappears, so it cannot evacuate chlorine. Chlorine accumulates, and you have problems. So the idea was simple. What if we gave diuretics to autistic children? Did you understand my reasoning? Chlorine level is high, valium has paradoxical level effects. We don't have any robust evidence that chlorine is high in autistic children, but it's true for epileptic children. Same with brain trauma and all that. We haven't checked that in autistic children. That's exactly what we shall do now. Now, Thalidomide has been used for a long time for babies. Bumetanide, main effects is to regulate potassium, because if you flush potassium too much, potassium goes down, the heart doesn't like it, careful with potassium. And so we started a program, and before handing over to Eric, now I've been working on a second thesis for 30 years. My thesis is that these diseases are ingrained or emerge around the time of birth. That's what I'm currently about to prove in autistic rats. This drop in chlorine levels doesn't take place in autistic rats either, because the oxytocin doesn't do its job or because the chlorine system is not regulated. In both cases, the time of birth is a crucial moment, even though it was initiated earlier. So this is our task force with the 3 musketeers, imaging, clinical trials.